1 in 8 women will be diagnosed with breast cancer in their lifetime. When detected early, breast cancer can often be successfully treated through surgery paired with chemotherapy and radiation. But despite advancements in breast cancer screening programs, in up to 10% of newly diagnosed cases, the cancer has already spread beyond the primary tumor in the breast to distant organs, including the bones, brain, and lungs. We call this metastatic breast cancer. Even when diagnosed early, up to 30% of breast cancer patients will go on to develop metastasis. There's currently no cure for metastatic breast cancer, and less than 1/3 of patients will survive beyond 5 years. My work aims to contribute to a greater understanding of the molecular mechanisms driving breast cancer metastasis through studying a protein called calpain. Calpain is found inside every single cell in the human body, and it works like a pair of molecular scissors to cleave or cut other proteins found within our cells. Calpain has over 100 protein targets involved in a wide range of cellular processes, including cell survival, cell death, and cell migration, or movement. All 3 of these processes are highly dysregulated in cancer, but I'll use the example of cell migration to highlight calpain's role. One calpain target involved in cell migration is a cluster of proteins known as a focal adhesion. And you can think of a focal adhesion like a parking brake in a car. Its job is to anchor the cell to its environment. Calpain functions to release this parking brake by cleaving the proteins that make up the focal adhesion. This weakens those anchoring points and allows the cell to move freely, much like a car once the parking brake is disengaged. Now we see the consequences of this parking brake-like action in our mouse models of metastasis. Using gene editing technology, I engineered breast cancer cells with no calpain in them at all, and I used these cells to establish tumors in the mammary glands of mice. I also made these cells fluoresce green in order to track how effectively they're able to metastasize or travel from that primary tumor to the lungs. The images you see on the slide are lungs taken from mice with mammary gland tumors, either with or without calpain. And as you can see, the cancer cells with calpain have a much easier time traveling to the lungs than the cancer cells without any calpain. In other words, those cancer cells with no calpain had their parking brakes engaged. Now you'll notice that the calpain disruption alone was not enough to stop every single cancer cell from metastasizing. Don't ask me how I know this, but it is still possible to drive your car with the parking brake on. Nevertheless, we hope to enhance existing cancer therapies by combining them with calpain inhibition. A greater understanding of calpain's role in breast cancer metastasis may lead to new combination therapies for patients who currently have very limited treatment options. Thank you.